Okay, so welcome to this next video in which we are discussing shear stress and endothelium derived relaxation factor. Okay, so I am trying to convince you that PCAM1 being phosphorylated is important for the activation of this endothelial nitric oxide synthase enzyme in response to increased shear stress. So we are going to basically do a gene silencing experiment using siRNAs. Okay, so siRNA stands for small interfering RNA. And basically, these are double-stranded RNA molecules that are very small. So here is our double-stranded RNA. So this is dsRNA for short. So ds means double-stranded. So this stands for double-stranded RNA. Okay, now in the case of small interfering RNAs, they are not very long. They will generally have around 20 to 24 24 base pairs. That's why the B was coming. I was just getting a little bit ahead of myself. Okay, so 20 to 24 base pairs. So 20 or so organic bases will be in this piece of double-stranded RNA. Right, so what we're going to do is we're going to produce a piece of double-stranded RNA that's small like this, which will have exactly a complementary code, basically, to the piece of mRNA that is going to make the PCAM1 protein. Okay, so let me explain this in more detail. On the level of the genome, you will have a gene which codes for PCAM1. So let's say this is the PCAM1 gene. Okay, now, this gene will be transcribed into a piece of mRNA for the PCAM1. Now, this mRNA has a certain sequence of organic bases, basically. What we are going to do is we're going to make a piece of siRNA that has the exact complementary sequence to this. And you might say, but look, we've got two of these strands. Okay, so what I would say is one of them will have the same code as this piece of orange DNA. So basically what we'll do is we'll take a small section of this, and I want to stress it will be a small section. I've blown it up here, basically. So these are the same barriers that I'm showing there, basically. So I've taken a small piece of this, which is around 20 to 24 base pairs long. We'll make an exact replica of it, and then we'll make the complementary strand to that here. So this is the complementary strand to that PCAM1 piece of mRNA there. Okay, now when we put in this small interfering RNA, what will happen is it will associate with a protein complex. Okay, so let me show you this happening. So here's our small interfering RNA, and here's our protein complex that has come and associated with it. Right, and this protein complex here, this is known as the RNA-induced silencing complex. So the RNA-induced, and that should have a little dash between it, RNA-induced silencing complex. Now this has a rather cool acronym. It's known as the RISC. Okay, I love that game. Right, R-I-S-C, except not spelt properly. Right, so this is the RISC here that has come and associated with our piece of small interfering RNA. And at the moment, the piece of small interfering RNA is still a double-stranded piece of RNA, so it's got both of the strands. Now what will happen is that the RISC will throw away one of the strands. Now, essentially it's arbitrary which one it throws away, so some RISCs, we won't just insert one piece of siRNA, we'll insert loads of them into the cell, and some of the RISCs here, they will throw away the orange strand, and some of them will throw away the green strand. We're not interested in those ones that throw away the green strand. They're not interesting. We are interested in those RISCs which throw away the orange strand. So now what's going to happen is this RNA-induced silencing complex, this RISC here, is going to just be bound to this green strand here. And now, what's going to happen is that green strand is now going to go and bind to the PCAM1 piece of mRNA. So what's going to happen is if I pull this PCAM1 piece of mRNA out down here, so here's our PCAM1 piece of mRNA, what's going to happen is this small, now we're drawing this in scale, here's this small little siRNA, and I'll still draw the risk larger, because otherwise it will look silly. Okay, here's the RNA-induced silencing complex, which is still associated with this little piece of now single-stranded RNA. So this is now a single-stranded, or SS RNA, to stand for single-stranded RNA. Okay, and here is the risk in blue here. Okay, so what overall has happened is that when you put in these small interfering RNAs, these siRNAs, they are being broken down into single-stranded pieces of mRNA, and one of those single strands is complementary to a certain portion of this piece of mRNA which codes for the PKAM1 protein. Okay, and now what's going to happen basically is that the risk is going to break up this PKAM1 mRNA into loads of pieces. So it's going to start chopping it up into loads of different pieces, and that's going to completely destroy the PKAM1 piece of mRNA. So is that PKAM1 piece of mRNA going to be translated and turned into a protein? No. So you're not going to get any PKAM1 protein, even though you're actually producing the mRNA, because the mRNA is being destroyed post-transcriptionally. Then it's not being translated and you're not actually making the protein. So this is a way of stopping those HUVEC cells, this human umbilical vein endothelial cells, from being able to actually produce PKAM1. Okay, so this is known as, this is a post-transcriptional gene silencing. Post-transcriptional gene silencing. Okay, right. And basically, when you do this experiment to the HUVEC cells, you knock out PKAM1, what you find is that eNOS no longer gets phosphorylated. So if you, well, so let me complete the story. What you find is that if you increase the shear stress now, when you've knocked out PKAM1 using this post-transcriptional gene silencing approach, you don't get phosphorylation of eNOS, and the amount of nitric oxide you produce is reduced. It's not gone, because the other pathway, remember, the other pathway of activating eNOS through the calcium calmodulin is still there, but the phosphorylation is gone. Okay, so that's what shows us that PKAM1 is upstream of this phosphorylation of the serine residues of the eNOS, this gene silencing experiment here. Okay, so we'll call it there for this video, and we'll continue our discussion in the next video.